Hi, everyone, and welcome to today's lecture on Mendelian genetics. Now, I do apologize because the part one and part two of the Mendelian genetics lecture has a lot of calculations and mapping things out, drawing things out. So, what you'll notice is I do take my time to go over every step of things like the monohybrid, the dihybrid cross, doing trihybrid probability calculations and so forth. But I also then, after that, put a slide for chalkboard notes in which I have all of the final drawn out notes from that explanation. So if you're one of the students who is able to just look at the notes and, figure it out from there, then I don't mind if you skip, let's say, the 10 minutes that I'm talking about one of the calculation type things that I'm drawing out in a lot of detail, okay? But I did still include it just in case for some of the students who feel more comfortable going step by step and hearing it all explained, okay? So that's why the Mendelian genetics part one and part two are going to be quite a bit longer when you look at the video lengths than some others. But again, I don't mind if you skip through to the chalkboard notes for those long explanations of calculations if you're someone that understands it. Now, I want to do a little bit of background in terms of Mendel's experimental method before we get into the details of this lecture. Now, most of you have already encountered information on Mendel, and so you usually know that he was experimenting with pea plants. That was an optimum model organism back then and still is. Some of the reasons why is when you look at them, so let me point it out here, when you look at the pea plant type of analyses, you'll notice that there are 7 clearly visible traits overall. And so when you have the visible phenotypes that have more than one variation, that makes using them very good. They are also easy to crossbreed. They have very quick generation times, so you can go through the various levels of heredity very quickly. And they're also cheap and, have minimal kind of requirements for working with them. Now, the first type of cross that I want to go over, again, you've probably encountered this in other classes, so you may skip over some parts, but this is the monohybrid cross. Mono means one, so you're only looking at one trait, and hybrid means heterozygous or more than one um, version or variation of that particular trait. So for instance, you might do a monohybrid cross looking at eye color only, or a monohybrid cross of Drosophila, which are fruit flies, looking at their wing length or wing type. Now, when we talk about crosses, there's some terminology I want to remind you of and for you to know. When you see P1, that is the parental group or the parental generation. It will usually be homozygous true breeding stock uh, for, let's say, the monohybrid that you'll set up in a laboratory. Then you have F1, which is the filial one. They are the offspring of your parental P1 generation. You then cross F1 flies or F1 organisms with the other F1 organisms. I know you're thinking, ew, that's kind of incest, but whatever, science. And when you cross F1 with F1, you make the F2, which is the filial 2 cross. Now, so this is mapped out for, let's say, when you're doing this in the laboratory, you start off with homozygous true breeding stock, but sometimes you can be given an individual cross where you're just told exactly which two flies you are crossing. And those can become the P1 or the parental of a cross in a question. Now, with monohybrid crosses or any kind of cross, you can predict inheritance using Punnett squares. And I want you to be very familiar with looking at a cross and being able to determine the expected genotype ratios. So those would be the alleles, for instance, seeing a plus or for representing wild type allele if you're looking at fruit flies, or a lowercase a, for instance, for apterous wings, All of this is terminology that we're going to see in the upcoming slides, and you'll also deal with in the laboratory. Okay, so now I'm going to give you an example of a monohybrid cross, and I'm going to draw it out in a lot of detail. And like I said, if it is too long for you and you remember this from other classes, you can then skip to the chalkboard notes right after it, where I have the final map or drawing of everything that I go through in the details. Okay, so like I said, the first thing that you're doing is you're starting off with the parental generation. And in the monohybrid cross that we were just looking at, okay, what did you have? You had homozygous wild-type fly crossed with homozygous recessive. Okay, so you can remind yourself that, that's one parent crossed with the other. This is the wild type parent crossed with the apterous parent. Now, when that parent goes to make its gametes, meaning its sex cells, the first parent, what can it give as a gamete? It only has one type of allele. It's homozygous wild type. So it has to give the offspring the plus, right? The wild type. What about the second fly? It only has apterous alleles. So it has to give that offspring an apterous allele. So parent number 1 gives a wild type allele. Parent number 2 gives an apterous allele. So that's the genotype for wings. Again, remind yourself we're dealing with wings here. So this fly offspring, what do we call it? That is now the F1 generation of flies. Okay, all of the F1 have the genotype of plus A. So that's the genotype of the F1 is plus A. And the phenotype, what's the phenotype for all of those? Well, as soon as you see that wild type allele, wild type is dominant. So they all have wild type long, normal looking wings. Now what you want to do to make the F2, you cross 2 of the F1 flies together. So you cross one heterozygous F1 fly with another heterozygous F1 fly, okay, in order to make the F2 generation. And the way you can predict what the F2 generation is you make a Punnett square, okay? And what you do is you have one parent's gametes up top here, and then the other parent's gametes on the side here. Okay, so you can remind yourself parent 1 and parent 2. Now with the law of segregation, remember, when a parent goes to make gametes from its genotype, those alleles separate for that trait, right? So what alleles can it give to a gamete? Well, the first gamete can be wild type. The second gamete can be apterous. Okay, so that's parent 1's possible gametes. This one over here will be parent 2's possible gametes. Now, what can our parent 2 give? Well, it can either give a plus symbol or an A, right? So a plus symbol or an A. Now all you do is you go square by square, combining parent 1's gametes with parent 2 gametes. So in this square, parent 1 is giving a plus symbol wild type. Parent 2 is giving plus symbol wild type. In the next box, you have parent 1 giving letter A and parent 2 giving the wild type plus. Now we usually write wild type first in the genotype, so I'm going to put plus and A. Okay, notice that each square represents the F2, the new baby, I'll call it a baby, the new offspring's genotype. And for that trait, we're looking at one trait because it's monohybrid. That trait, you have to have two alleles, one from mom, one from dad. So now we finish out the bottom. Parent 1 in this square is giving an allele of wild type. Parent 2, an allele for Apterus. And the last one, both parents are giving an allele for Apterus. Okay, so the F2 generation now, you notice F2, sorry, my pen does not want to keep writing. It's dying over here. Okay, so our F2, you notice that there are 4 offspring in that Punnett square. So the F2 genotypes, we're going to write first. You notice you have the first square had plus mark and plus mark. Both of these middle two had plus A, okay? And then the last option was AA, okay? So you had 1 out of the 4 were homozygous wild type, 2 out of the 4, or 1/2, was the hybrid or the heterozygous, and then 1 out of the 4 was fully apterous, okay? So the genotype ratio was 1 to 2 to 1, okay? And you could write the genotypes underneath to remind yourself what genotypes you're referring to. Then you also have to keep in mind the F2 phenotypes for the monohybrid cross. Okay, so what does plus plus look like? Well, that looks like wild type wings, right? But what else looks like wild type wings? Plus A, right? The heterozygous, because as soon as you see any plus mark, it's going to look wild type. So now you have 3 out of those 4 boxes, 3 out of those 4 options look wild type. So all of these guys here will look wild type, whereas the only one that will look apterous because it has fully apterous recessive alleles is the last one. So 1 out of 4 will look apterous. And remind yourself, what does apterous look like? No wings, okay? So 3 out of 4 phenotype-wise will be wild type. 1 out of 4 will be apterous. So the phenotypic ratio is 3 to 1, wild type to apterous, okay? If you have any trouble following the P generation, the F1 or the F2 generation, please let me know. I could ask you to cross the flies. I could ask you about the genotypic ratio. I could ask you about the phenotypic ratio in a monohybrid cross. Okay, if you'd like any examples or practice problems, please contact me in the Remind app to let me know. So now with Mendel's research and his analyses, he came up with the first 3 postulates that you see here. The first one is that unit factors occur in pairs. Now what I want you to do is cross out the word factors and replace that with the word alleles. So later on we started calling them alleles. Now why do these alleles occur in pairs? Well, you get one from each parent for each particular trait that you looking at. So when you're looking at a trait and you have 2 different alleles, you got one of those alleles from mom and one of those alleles for dad. Now, when we talk about alleles, you also have the concept of dominance and recessiveness. Okay, so dominance, we're not talking about alpha bros here. Dominance means that when there are two different alleles for a particular trait, such as, let's say, eye color of the fruit flies, one of those alleles will be dominant to the other. Okay, so when you have a dominant allele, if it's fruit flies, we will show it as a plus symbol. If it's any other organism, we usually give it the capital letter. Okay, we then give the recessive allele a lowercase. Dominant alleles means that once you have at least one dominant allele present for that trait, your phenotype is going to look like that dominant trait, that dominant allele's phenotype. So the dominant allele will mask the recessive allele. So the only way that you'll see the recessive trait is if you have two of the recessive alleles and you don't have any dominant alleles present. The last postulate that he showed here is segregation. And so with segregation, that's what you see in this little figure here. It's the concept that when you are making gametes, meaning sex cells, either sperm or egg, the alleles for a particular trait will separate randomly so that each gamete will then receive one or the other allele with equal likelihood. Now we get to a concept that I love. So circle star highlight the concept of the test cross. So a moment ago, I mentioned that there are two different types of alleles. There's the dominant one, such as the capital D here, and then the recessive allele, which is the lowercase d. Now, the only way that you can see a recessive organism is if its genotype is only recessive alleles. So that means if you see the recessive trait, such as dwarf, you know the genotype right away because you could only have dwarf if there's the two lowercase d recessive alleles. Now, if, however, you see a tall organism in this scenario, you don't know, is it homozygous, dominant tall alleles, or is it heterozygous with the dominant tall allele masking or hiding a little recessive allele in there? So that's where the test cross comes in. A test cross is used when you have a dominant looking organism and you want to figure out, so dominant looking phenotype, and you want to figure out its genotype. Is it homozygous dominant or is it heterozygous? Okay, so that's the purpose of a test cross. The way that it is done is you cross that homozygous looking, you cross that dominant looking phenotype organism with a fully recessive organism. Okay, so if you see it looks dominant, the way to tell its genotype is to do a test cross, meaning you cross it with a fully recessive organism and look at its offspring. If you see that they are all dominant looking, which in this case is all tall, that tells you that the unknown organism's genotype was homozygous dominant. If you do the test cross and you see some dominant looking, some recessive looking, that tells you that your unknown dominant genotype was heterozygous. Now we are going to review the dihybrid cross. This is different from a monohybrid because, well, think about it. Mono means one, so you're only looking at one trait. Di means two, or what my soul does a little bit more and more every day. But anyway, so a monohybrid is only looking at one trait, whereas a dihybrid is going to look at two traits. So Mendel in his dihybrid looked at seed color and seed texture at the same time, for instance. In your genetics laboratory class, you usually look at the Drosophila or fruit fly eye color and wing type at the same time. So two different traits at the same time. Now, even though it's a bit more complicated, you can still track a dihybrid with Punnett squares just like you could with a monohybrid. Now, when you are doing this, it looks really scary. We're going to go through it in the next slide step by step. But what's important to keep in mind, each gamete that you're going to have in that cross, in a dihybrid, each gamete must have one allele for each trait. So for instance, if you're doing the fruit fly experiment with a dihybrid, each gamete should have one allele for eye color, one allele for wing type. That way, when you cross the two gametes together during fertilization, each offspring ends up with two alleles for each trait. They get one allele from mom, one allele from dad. Okay? So just keep that in mind when we're going through the very big and scary dihybrid cross in the next slide.
Okay, so now again, keep in mind for dihybrid, we're going to be focusing on two traits now. So it might look a little scary, but it's actually quite easy if you keep everything nice and organized. Basically, keep reminding yourself that you're looking at two traits. Now, because it's a dihybrid, again, you want to be crossing two hybrid flies together, but just like with the monohybrid, you have to first make those hybrids, those heterozygous flies. So you start out with your P1, your parental generation, being homozygous stock flies, meaning stock, they're coming from the manufacturer. So you want to start out with homozygous flies, and we cross an apterous fly with a sepia fly. Now keep in mind that flies are named after their mutation, but we're looking at two traits. We're not just looking at one trait. So when we look at our first fly, when it's called apterous, that tells us that the wings are mutated. But what's our second trait that we're looking at? We're looking at eyes as well, right? Well, do you see any word for eyes in the name apterous fly? No. So what does that tell you about its eyes? Its eyes are wild type. Okay? So when you draw out the genotype for this apterous fly, you would first draw out the homozygous wild type genotype for the eyes. And then you would write the alleles for wings, which in this case for apterous would be two lowercase recessive alleles. So again, notice that that genotype has 4 alleles because we're dealing with two traits. Each trait, so eyes and wings, each have 2 alleles because that fly got 1 allele from mom, 1 allele from dad for each trait. Okay? So then we cross it with our homozygous sepia fly. Now sepia describes the eyes, right? So mutants are recessive homozygous mutant for the eyes. What about the wings? What would the wings be? Well, do you see any word for wings in the name of that fly? No, it's just sepia for the eyes. So that tells you that the wings are wild type. Okay, so just like we said before, you have your two traits, each having 2 alleles. Now, when you go to make the F1 generation from those parent flies, well, what do you notice? Again, think of the traits separately. So for eyes, these offspring can only get a plus or a plus. So no matter what, the F1 will get 1 plus allele from parent number 1. And what eye color allele can it get from the second parent? Either lowercase s or lowercase s. Either way, it's getting an S as the second allele. Okay, then you do wings, the second trait. So what allele can it get from parent number 1? Either an A or an A. So it's getting an A from parent number 1, and from parent number 2, it can only get a plus or a plus. And again, we usually write the plus symbols first ahead of the mutant allele. Okay, so again, keep going based on each trait. So all F1 will be completely heterozygous. So there'll be heterozygous, 2 different alleles for eyes and for wings. Okay, And so you cross the F1 with each other. Again, I know gross, some of them are siblings, but that doesn't matter to flies, okay? And then you move on to see what kind of F2 this heterozygous cross can produce. And so to do that, we're going to make a big, scary looking Punnett square. Okay, the dihybrid Punnett square looks very scary, I know, but again, it's very doable if you keep it nice and organized. Okay, so we're going to make a big Punnett square here. Okay, and we're going to separate it based on the different gametes that each parent can give to the offspring. Okay, so we've got to just draw this out first. We're going to have parent number 1's gametes over here, and then parent number 2 along this side. Okay. All of the things we're going to write now will be the 4 gametes possible for parent number 1 to make. Okay, so in this top area, we're going to put the gametes or the sex cells that parent number 1 can make. Now, when parent number 1, the apterous fly, the heterozygous fly goes to make its gametes, when you make gametes, sperm or egg, what do you know about your alleles? Well, they separate, right? We did that as the law of segregation last time. And so each gamete that this parent makes must have 1 allele for eyes and 1 allele for wings, right? So we're looking at this parent. What gametes can it make? Well, 1 allele for eyes, 1 allele for wings. So the first gamete is plus plus. Its second gamete is again 1 allele for eyes, 1 allele for wings. So 1 allele for eyes, it could give a wild type. Second, now we go 1 allele for wings. We already have the first gamete plus plus, so this one instead can give little a. Then that fly can also, for eyes, if it doesn't give a wild type allele, what does it give? It gives its recessive allele. Okay? And then for wings, it can either give wild type or A. So we'll give wild type here for wings. And then the last one, eye and wing, it can give its recessive, or for wings, it can also give recessive. Now you'll notice that our F1 parents, parent 1 and parent number 2, so this is parent 2, they have the same genotype, so they can make the same alleles and the same gametes. So parent 2 will also make 1 gamete that's plus plus, 1 gamete that's plus A, 1 gamete that's S plus, and 1 gamete that is SA. Okay? Now again, normally you would put plus in front of, the recessive, but in this case, we're going to keep them organized as eyes and wings. Okay, so that you remember, gametes are 1 allele for each trait. Now you put the gametes together. So this first gamete fuses with this second gamete, so parent 1's sperm, for instance, fuses with parent number 2's egg over here, and now the offspring will have 4 alleles. And the way you figure out what's what is you combine the eye alleles first and then the wing alleles. So eyes have a plus and a plus. So that's plus plus. What do the wings have? Plus plus. And you do that for each one. You do eyes and then wings. Okay, so you keep them separate. Eyes are plus, plus, and wings now are A and a plus. Okay, So now in here, I'm going to make sure to keep the plus symbols in front of their recessive, but you know where the A and the plus came from. Okay, and you would keep doing that for every single one. You would do eyes, wings. Okay, so we have plus S and plus plus. Eyes are plus S and wings are plus A. Okay, and we're going to do that for each one. I'm going to stop writing all the extra stuff and just fill them in. If you get confused at all, just send me a remind message with a picture of this slide, and I'll go over what I just did here. Okay, so each one I'm putting eyes first and then the wing alleles. Eyes first, then the wing alleles. Eyes and wings, eyes and wings, and then we do at the bottom eyes, whoops, wrong, sorry, my pen erased that. So eyes and wings, eyes and wings, eyes and wings, and the last one, eyes and wings. Okay, like I said, if for any of them you're uncertain where any of the alleles came from, why they look the way they do, just please send me a message and remind, and I will clarify where everything came from. Okay, so those are all of our genotypes. The last thing we have to do, so you notice there are 1, 2, 3, 4 times 4, you have 16 different little squares here with all of the genotypes. And then what we do is we map out for the F2 the different phenotypes. And again, phenotypes are what you see. Okay, so you can see 4 different types of flies when it comes to a dihybrid. You can either see a fly that has wild type eyes and wild type wings, okay, or you can see a fly that has wild type eyes and mutant wings, which we'll write as APT for apterous wings, meaning it has no wings at all. Or you could have the mutant eyes. So you can have sepia eyes with wild type wings, or sepia eyes with mutant wings. Okay, and so we're going to look at our little squares on this Punnett square. We're going to look at the individual little squares to figure out which ones have wild type eyes, wild type wings first. Okay, so we have 1 wild type eyes, wild type wings. What we're doing is we're looking for any of the ones that for both traits have at least 1 plus symbol, because that's dominant. 2, 3, 4, all of these, notice each trait has at least 1 plus symbol. Okay, so that's 4 wild type. This one as well. The next one we won't circle because it's got 2 apterous alleles. Then we're back to, again, 1 plus and 1 plus there. Another one that's wild type for each trait, wild type for each trait, wild type for each trait, and then all the rest have some recessives. So then we count up the ones that we circled. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9 out of our 16 little squares are fully wild type looking. Okay, and then next we're going to look for the ones that are wild type eyes and apterous wings. Okay, so we're going to look for any of the ones that have double little a's for the wings, and for the eyes, it has to have at least 1 plus mark. Okay, so what do you notice? Those are 3 of them that had the wild type eyes and the apterous wings. Then you look for the flip or the reverse of that, the ones that have the 2 little S symbols, so they're sepia eyes, and they have at least 1 plus mark. Okay, at least 1 plus mark, so that would be 3 of them. Okay, so we have 1, 2, 3 of them with sepia eyes and wild type wings, and that leaves us with our 1 out of 16 fully recessive mutant. Okay, so that's why when we talk about dihybrid crosses, meaning crossing 2 fully heterozygous flies or organisms for 2 traits, you will see a 9 to 3 to 3 to 1 phenotypic ratio. Okay, and again, phenotypic being what you're actually seeing for those flies. Okay, now that's how you do the Punnett square, but in the coming slides, I'm going to show you a little easier way to figure this out based on considering a dihybrid as simply being 2 monohybrids put together. Okay, again, if you have any questions or concerns, send me a message in the Remind app.
Okay, so now that I taught you the brutally torturous, boring way to do a dihybrid cross via Punnett squares, I wanted to show you that thanks to some of the other postulates, you could actually figure out dihybrid cross or even trihybrid and onward cross probabilities of their possible genotypes based on a fun little thing called the product law of probabilities. And we're going to show this in a few slides from now, how to actually use the product law to calculate these outcomes. Basically, what it is that when you have different events occurring at the same time, the probability of those outcomes can be figured out based on multiplying the individual probabilities. So I'm going to show you in a minute how you can figure out the probabilities of each dihybrid cross outcome by actually treating them like monohybrid crosses and multiplying together the individual probabilities. One other postulate that I want to show you that's part of the dihybrid cross is the law of independent assortment, which is shown by this little figure here. It basically shows you that even though you have all of these alleles and you have more than one trait for the dihybrid cross, one trait does not influence the separation of the alleles into the gametes for another trait, meaning that you could end up with multiple different variations of the alleles going into gametes, okay? So for instance, when you look at these parental genotypes, when they're making their gametes and then you have the heterozygous, the dominant alleles, for instance, for each trait do not have to stay together when they're going into the gametes. It's independent assortment. All of the alleles for the different traits are able to assort separately into their final gametes. So now here you see an example of using the product law for the dihybrid cross. And when you look at this, we did the dihybrid cross in the big Punnett square example. So you know that 9/16 of the offspring are expected to be fully wild type. Instead of doing a Punnett square, you could just multiply together the monohybrid probabilities. So the probability of getting a wild type offspring for eyes is 3/4. The probability of getting a wild type offspring for wings is 3/4. And so you can just multiply these two together to obtain that 9/16. The way that we get the 3/16, for instance, for sepia wild type is because the monohybrid probability of getting a sepia, which is the recessive trait, is 1/4, and the probability of wild type is 3/4. if you're looking at monohybrid crosses, so for eyes and for wings. And if you multiply them together, that gives you 3/16. And we're going to go through this in more detail when I show you in a few slides from now how to use the law of the product rule in terms of the trihybrid cross. Now, one other thing that I want to mention about a dihybrid cross is that the test cross that we mentioned previously can also be applied to dihybrid crosses. So just as a reminder, what is a test cross? Well, that means that you see visually a phenotype that looks like the dominant traits. So in the case of a dihybrid, let's say you were looking at eye color and wing type, you would see a fully dominant appearance, something with wild type eyes and wild type wings. Now, you would not know, since dominant alleles can mask recessive alleles, you wouldn't know the genotype of that dominant looking organism. So you would do a test cross. The way that you would do that is you would cross the fully wild type looking fly with a fully recessive fly. Okay, so in this case, it would be sepia apteris. Sepia is recessive. In this case, for eye color, apteris is the recessive trait for the wing type. And you would look to see what type of offspring you have. And so I'm going to show you all written out and drawn out the various options of what offspring you might find depending on what the genotype of that unknown wild type fully dominant looking fly is.
Now we're going to draw out what we would actually look at or look for in this exact test cross. So just got to get the pen ready. So there are 4 options when you look at this test cross. You have option 1. Okay, with option 1, if that fully wild type, okay, so fully wild type, you want to know what genotype. So you're crossing it with a sepia apteris fly. So option 1 is if that fully wild type looking fly has a genotype that's all wild type alleles. We'll write it out this way, and remember to always think of it as eyes, wings. That's why you have those 4 alleles, because 2 of them are for eyes, 2 of them are for wings. Now what you're doing is you're crossing it with the sepia apteris fly, and if that is what your wild type fly is, then what do you notice? For every trait, no matter what, this first fly is going to give a wild type allele. So for eyes and for wings, that unknown fly will give a wild type allele. The other fly for eyes will give sepia, and for wings will give apteris. Okay, so whenever you write out your 2 traits, and then write out the alleles from parent 1, parent 2. Okay, parent 1 for wings, parent 2 for wings. Okay, and that will help you keep everything organized, figure out what's coming from what. Now there's another possibility. Maybe that wild type fly is, sorry, that was supposed to be an arrow. Maybe that wild type fly is homozygous for eyes, but heterozygous for wings. Okay, and the way you'll know this is when you do the test cross, what kind of offspring will you get? Well, some of them will inherit the wild type, both wild type, from that parent. Okay, so again, we're doing eyes, wings. So some of the offspring will get wild type from parent 1, wild type eyes, wild type wings. And then from parent 2, what are they going to get? No matter what, S and an A. Okay. What else what other flies can happen? Well, parent 1 can only give wild type for eyes, but look, it has 2 different options for wings. So what if parent 1 gives wild type for eyes, but for wings it gives Apterus? Well, no matter what, the second parent is going to give S and A, okay, for eyes and wings. So what happens in this scenario, half of the flies will look fully wild type eyes and wild type wings. Half of the flies will have mutant wings. So since the eyes are wild type here and the wings are mutant, we would call this fly Apterus. Okay, so half the flies are Apterus, half the flies are wild type with this option when it's heterozygous for wings. If instead it's heterozygous for eyes, then you'll know that because in the results you'll suddenly have half wild type but half sepia. And so we're going to draw this out now. If it's heterozygous for eyes but homozygous now for wings. Okay, so in the second option, it was homozygous for eyes, heterozygous for wings. Now we're going to look at what if it's the opposite. Again, it's a test cross, so it's always crossed with the SSAA fly. And in this result, half of the flies that result from this cross will have plus from parent 1, plus from parent 1 for wings. And then the other allele is always S, A, because that's all this guy can give, okay? And half of the flies, if they're not getting the plus for eyes, they're now getting the lowercase s from that parent, okay? So now it's giving S, and for wings, it can only give a plus, okay? So we're looking again, that's parent 1, and then parent 2 always gives S and an A. And what did I say before? That this half of the offspring will end up being sepia. Okay, so half wild type, half sepia. Then the last one is option 4. And in option 4, what if that unknown wild type fly is fully heterozygous, meaning eyes are heterozygous and wings are heterozygous? Well, what will the offspring look like now? Well, you keep doing what we've been doing. Okay, you're crossing it with that fully recessive fly. In the first set, again, we're doing eyes, wings, and what do you notice? For eyes, you can get wild type from that parent. For wings, you could get wild type from that parent. Okay. No matter what, parent 2 is always given S and an A. Or what else can you get? For eyes, you can get the plus, but then the parent 1 can give wings, being Apterus. Okay, so parent 1 can give a plus and an A gamete. And what will the second parent give? Always S and A. Okay? Or there's still more. What if instead parent 1 is giving the sepia mutant allele? Okay, so for eyes, if it's giving sepia, it could then for wings either give plus or A. So plus, again, the second parent always giving S and A, or it can give that sepia allele, okay, for eyes, but now instead it can also give the Apterus allele. And once again, this second parent is always either giving S or A. So that's what you end up as the last option. So notice for the fully heterozygous wild type, you get 4 different options, okay? You'll end up with seeing a big variety of flies. So you have 4 different options. 1/4 of the offspring will be fully wild type. 1/4 are apterous. 1/4 are sepia. And 1/4 are the full double mutant, sepia apterous. Okay? So basically, when you do a dihybrid test cross, ask yourself, do you see all the offspring are wild type when you cross that unknown wild type fly with a fully sepia? Are half of the offspring wild type and half apterous? Are half wild type and half sepia? Or do you get all 4 variety? Okay, if you're capable of getting all 4 variety, that tells you you have a fully heterozygous wild type fly. If you're only seeing one of the mutants, that tells you that for that trait you are heterozygous, but the other trait, your fly is fully homozygous wild type. So in this case, you notice only the wings are heterozygous, so you suddenly see apterous flies. If you're seeing sepia flies, well, that tells you only that eye trait is heterozygous. And then, like I said, if all 100% of the offspring are wild type, then you know that you have a true blue 100% homozygous fly wild type for both traits. Okay, so I know this is a lot of information. If you have any questions on this test cross, just let me know. I could give you some other examples, but basically be aware of what does it mean when you're doing a test cross. Why are you doing this test cross? What are you doing with it? And what are some of the options, okay? And keep in mind, like I said, this can be done for monohybrid or for dihybrid.
Now, when we were talking about dihybrid crosses, I very quickly and briefly introduced you to the law of probability, but I mentioned that we'd go into it in more detail in later slides. So what I'm going to do is on the next slide, I'm going to show you drawn out how we can use the law of probability for something like a trihybrid cross. Just like I mentioned, we could use it for a dihybrid cross. We could go even further and put 4 or 5 traits, but I'm usually not that mean. We'll stick to the trihybrid for now. And I'll show you, even though this figure shows you the branched method that can work, the easiest thing to do is the law of probability. And we're going to go through that in the next slide. And make sure to circle star highlight that because it makes for a great question on an exam where I could come up with any trihybrid cross, just mixing up capital and lowercase letters, and ask you for the probability of a particular type of offspring.
So let's say you are looking at a cross of capital A, lowercase a, capital B, lowercase b, capital C, lowercase c as one parent, and it's crossed with capital A, lowercase a, so dominant recessive, dominant B, and dominant C. So you have 2 organisms, you're looking at 3 different traits. And I could ask you what fraction of the offspring are predicted to have the genotype of capital A, capital B, lowercase b, and capital C. And the way you would do this is look at the 3 traits separately as if they're monohybrid crosses. So you can make 3 different Punnett squares, okay? So the first Punnett square would be the parent that's capital A, lowercase a, crossed with the first trait of your second parent, who's also capital A, lowercase a. Okay, it's a regular monohybrid cross, so you've seen this kind of ratio before. Okay, then you make a second Punnett square for the second trait, which in this case you have heterozygous for B crossed with homozygous dominant for B. Okay, so you'll have capital B's over here and heterozygous over here. And then you have a third trait, so you do your third Punnett square for the heterozygous C and the homozygous dominant C. And again, it looks very similar to your second Punnett square. Now we want to know what fraction of the offspring are predicted to have this genotype here. So first we ask ourselves, what fraction are capital A, capital A? Well, that's 1/4. What about what fraction of the offspring are predicted to be heterozygous for B? That's 1/2. Okay, because it's 2 out of the 4 squares. Then we have homozygous dominant for C. And again, that's also 1/2 of the Punnett square is heterozygous for capital C. So now all you do to get this predicted genotype is you multiply your 3 probabilities. Okay, the individual probabilities, the probability of AA, 2 capitals, the probability of the heterozygous B, and the probability of the homozygous dominant C. And you get 1/16 for this genotype. I could then instead ask you probability for heterozygous A, and then the remaining traits that we had above. Okay, and you would do it in the same way, except now, what's the probability of heterozygous in our first Punnett square? 1/2 of that Punnett square is heterozygous for A. The other values stay the same. Okay, 1/2 of the second Punnett square is heterozygous for B. 1/2 of our third Punnett square is homozygous dominant. So now the probability for this genotype would be 1/8. Okay, and I'll write out that genotype as well. Okay, so as you see, you can do as many. I could have even added a 4th, a 5th, a 6th trait, and all you keep doing is looking at each of the traits individually as a monohybrid Punnett square and just multiply the probabilities together. Again, if you have any questions, any concerns, you want some more practice problems, please just send me a message in the Remind app. You could even send me a picture of the particular slide that you need a little extra help with.
The last thing in part 1 of this lecture that I want to mention is that eventually when they discovered chromosomes, it actually matched up with the concepts that Mendel had described in things like his postulates when he was just talking about things like factors which became alleles and whatnot. So chromosomes ultimately kind of proved that hey, he was right in what he was thinking. And so this figure is just showing how chromosomes demonstrated things like independent assortment once the chromosomes were actually discovered. Okay, now that was a ton of information, and again, I'm sorry that Mendelian genetics tends to be some of the longest of the lectures because I do go step by step to draw out the different Punnett squares and calculations and whatnot. Now, what I want you to do is take a look at these 2 questions here, and please send me your answers along with a picture of this slide. Please, please, please always send me a picture of the slide too, so that I know exactly which question you are answering. And so send me your answers to both of these as soon as you get to this in the video. I don't care if it's 1, 2, 3 o'clock in the morning. I have no life and it won't wake me up if you send it at that time. I just don't want you to forget to send me these answers. It's very important for me to then be able to help and make sure that you truly understand the concepts. Okay, so now I want you to take a nice break before you then move on to part 2 of the Mendelian genetics lecture, as it is a lot more calculations. I am so sorry, but genetics is my baby, and so hopefully I can help you to love genetics as much as I do, even when there are calculations.